years, I have had the opportunity to work with a plant called Arabidopsis thaliana. Arabidopsis thaliana is a small plant related to canola. This is a good plant to use for scientific research because it has a very short lifespan. The entire genome has been sequenced, and there are many different known mutations of the genome that produce different effects on the plant when exposed to different environmental factors. The one that particularly interests me is exposure to chemicals through the soil, and the effect of that exposure on the germination rate of future generations. This is capable of being measured due to the fact that Arabidopsis plants are self-pollinating, meaning that no other plant's DNA is capable of getting into the seeds of one plant. The next generation is essentially the clone of the plant before, including any changes in the plant caused by exposure. The chemicals I worked with were caffeine, cadmium, and nickel sulfate hexahydrate in particular. Cadmium and nickel sulfate are both found in soil, especially in and around the Midwest. That is part of the reason why I picked those two to work with, since they affect the area that I live in the most. This procedure included learning many things about plant biology, molarity, microscopy, and aseptic technique, as well as being able to use Excel for calculating percents and creating graphs. The plant biology knowledge required included being informed about the different parts of the plants, like sepals, true leaves, calyx, and flowers, in order to take measurements of many different variables. For the nickel sulfate, a 4 millimolar solution was needed to be created, meaning knowledge of pipetting and serial dilution could also be useful. Microscopy using a dissecting scope was useful after harvesting the calyx in order to separate the seeds from the other plant debris. After collecting the seeds, they were sterilized using a solution of water, bleach, and twine, and then plated on an agar plate using aseptic technique to ensure the only growth on the plates was the Arabidopsis seeds. The aseptic plating took place in a biosafety cabinet, which not only has a curtain of air to keep the samples from getting contaminated, but also one to keep the technician safe from the samples. When I worked at Mizzou with the plants, I used a laminar flow hood, but in my high school lab, the biosafety cabinet was the only option. The plates were square and had a 6x6 grid on them, a total of 72 seeds on each plate. After about a week, the seeds had begun sprouting. The percent of seeds that had sprouted was then found using Excel. This was done for all 6 different genotypes that I worked with, the main two being RLD wild type and SRFR1-1 mutant type. I find these plants extremely interesting because not only can they show how that particular plant will react to external stimuli, but also how many other plants will react as well. For 3 years, these plants have intrigued me and prompted me to try to find out more about them. Not many people know just how useful Arabidopsis thaliana is, but I hope that I can use these plants for experiments to help aid my learning in the field of chemistry in the future.